That's the thing to search for. It's very important for scientists to take vacations. It makes you a better scientist. And you can quote me on that. So, as always, we can start our class by relaxing. And not to worry about a thing. And let's take a nice deep, if you feel like it, a nice deep sigh of relief. We're at the fifth class. The first four classes we talked about one design principle that biological networks, transcriptional networks are made of a small handful of circuit elements that occur again and again and again, each one with the defined dynamical functions that we can understand, test experimentally. Let's take another nice deep sigh of relief. For the new people coming in, there's places here up front. You don't have to be afraid to sit here if you feel like it. And now we're switching to a second design principle. And that is the principle of robustness. So I'm just going to write down up here this design principle. And this whole class will be devoted to looking in detail at a biological system, bacterial chemotaxis. So we can have the pleasure of seeing a system in more detail than we did before. And we're presenting this system because it's a good example that demonstrates the general principle of robustness. So what's the principle of robustness? That biological systems are designed such that their important function is insensitive to the naturally occurring variations in its parameters. So this sentence we'll try to unpack it during this class. What it means is that if bigger letters, that if the biological system needs to do something precisely, it's faced with an inherent problem. The biological material has some randomness in it. This randomness comes from stochastic processes, random processes, because, for example, making proteins, to make proteins you start from one copy of the gene on the DNA, or two. There's two sister chromosomes. It's just a single molecular object. And then that passes through some RNA molecules, just a few. So you've passed through low numbers. And because of that, there's cell-to-cell variations in the concentration of each and every protein. That's just one example of naturally occurring variations in biological materials. Also, the environment changes, temperature changes, pH changes, etc. So that's also naturally occurring variation. And if your circuit or system needs to do something very precisely, it has a challenge. How to do something precisely despite the fact that it's built out of noisy substance that has in it unavoidable inherent randomness, and living inside an environment that has some unexpected qualities. And this constraint that we need to be insensitive to the natural randomness is very strong. It limits the number of possible designs that we can write down on paper to just a very, very few that can actually work in the real world. And that's very powerful for our theoretical understanding of biology, because it turns out that real biological systems, let's take another nice deep sigh of relief. The systems that have been studied in detail are built in very special, very elegant and beautiful ways to make them robust. So that's the principle of robustness. To demonstrate that, we'll talk about bacterial chemotaxis. These are bacteria, and they're swimming here, different directions. They're not worrying about a thing because they are able to do something quite remarkable. When the going gets tough, the tougher they get going, as they say, when conditions are harsh, they are able to swim away with their flagella, we talked about already. They're able to climb gradients, chemical gradients of food, and escape, go down gradients of toxins. And this property of moving up gradients is called chemotaxis. It means when you see a chemical gradient, you call for a taxi and you go up it. Tax means in Greek to walk, to move. This was discovered in the 1900s. And the simple experiment is you take a dish with swimming E. coli and you put a pipette in it that has, let's say, an amino acid that the bacteria can eat or sugar. They form a cloud. If you look through a microscope, you can see they form a cloud at the mouth of the pipette. And if you put in a pipette that has toxin, for example, nickel ions, something the cell doesn't like, they escape from there. Their bacteria, almost all species of bacteria have some form of chemotaxis. Some bacteria, not E. coli, can do phototaxis. They can go towards light, or some bacteria go away from light. There's bacteria that do magnetotaxis. They can follow the magnetic field of the Earth. They have inside them, they build nanometer-sized magnets of iron crystals. And they use that actually to tell where is up and down in the water, because the lines of the magnetic field go like that. And there's bacteria that do taxis to electric fields. Bacteria that live in silver mines can do silver taxis. E. coli can do chemotaxis to quite a number of different chemicals, as we'll talk about. So in the pipette, if I put in a chemical that draws E. coli, it's called an attractant. And if it runs away from it, it's called a repellent. Now, this E. coli here is a 1 micron sized animal. In order to do this chemotaxis, it has to overcome astounding physical limitations. So I'll tell you first of all the dynamic range, how sensitive is it to gradients. It can detect gradients so this size is 1 micron, right? Let's imagine a volume here around E. coli. It's about the same volume as the cell. So let's say 1 micron cubed volume. It can detect gradients so small that if there is 1 molecule of this attractant out of 1,000 difference between its head and its tail. So the concentration is 1,000 molecules per cubic micron. That's the concentration. And the gradient is so small, it's just 1 molecule difference on average from here to here. It can detect it. That's a gradient of about 1 nanomolar per micron over a background of 1 micromolar. So. And it can detect a 10 to the 5th range of gradients. So it doesn't become blind by bigger and bigger gradients. 10 to the 5th range of gradients. In fact, if you put in the pipette with 10 to the 5th, different experiments with a 10 to the 5th difference in the concentration of stuff you put in the pipette, the speed at which they accumulate is more or less the same. They go to the source about the same speed over 10 to the 5th range of concentrations. It's a very, very sensitive device. Now, consider that they do this being 1 micron sized animals, as I said, subject to Brownian motion. So, the water molecules hit them unequally on different sizes. That's very huge for micron-sized objects. So if they try to swim straight for 10 seconds, Brownian motion on average makes them change direction by 90 degrees. So they're under quite a lot of thermal noise. It's like walking in a very, very strong wind, you can say, for them. But of course, this is Brownian motion. They're too small to sense the difference between their own head and tail because of this tiny difference of, if it's 1,000 molecules in a volume, just the Poisson fluctuations there's an average, of 30 molecules plus minus. So to detect one molecule from head and tail, they're too small to do that. They don't have antennas at their head and tail. They can't do that. So how do they do it? Good question. And the answer was figured out in the 1970s by a physicist called Howard Berg that built a microscope with 1970s electronics that tracks the bacteria, just keeps it in the middle of the frame, and as it's moving a lot, it just follows it like that. By the way, you don't have to write down unless it helps you remember, because this is all, what I'm doing is from chapter 7 in the book. You can find in the library. So E. coli do chemotaxis using a biased random walk. They actually compute temporal gradients. So if you look under the microscope at individual E. coli cells, what you see them is swimming for about a second, then stopping for about 1/10 of a second, then swimming in a new direction, then tumbling, swimming in a new direction, tumbling. Doing a random walk. These straight parts are called runs. They last about 1 second on average. They have an exponential distribution of length. And these changes of direction are called tumbles. They last about 1/10 of a second. That's what they do, let's say, in a liquid that has no gradients in it. They search space like that. And if you look a little bit more close now at E. coli, at the cell that's doing this, you notice that it has flagella. It has about 6, 10 flagella. And each flagellum, we talked about, is connected to a motor. And this motor, as I told you before, is an electrical motor that spins at 100 hertz based on protons going into the cell. And it turns out that the way this works, the difference between runs and tumbles, has to do with the direction of rotation of the motor. The motor spins counterclockwise. The flagella align into a bundle that pushes the cell forward. And if it spins clockwise, the flagella fly apart, causing a tumble. So this motor has clockwise and counterclockwise directions. Now, how does E. coli detect a gradient? So let's imagine I put here the pipette that has an attractant. Some examples for attractants are sugars, amino acids like aspartate and serine. Some examples for repellents are amino acids, other amino acids like leucine or metal ions. Also, E. coli, by the way, does thermotaxis. It runs away from high and low temperatures and goes to the temperature where it grew up. So if it was raised in a flask at 37 degrees, it goes to 37 degrees. So if we put in attractants, the strategy of E. coli is, what it does is compare the present concentration of attractant to its past concentration. If this concentration increases, it decreases the probability to tumble. So this is a very simple rule. What it does is it looks at the concentration now and remembers the concentration in the past and compares them. And if things got better, then it stops tumbling. So that it tends to continue in the same direction. And so when you look at bacteria and you put the pipette there, you don't see much. It looks like a random walk. But if they happen to go in the right direction, there's a higher probability that they keep going there than if they're going in the wrong direction. And that's enough to guide a cloud of bacteria like this up the gradient. By the way, if there's a repellent, if the concentration increases, you increase tumbles. And by that way, if you're going up the repellent concentration, you change direction randomly. It's a random change of direction. It's not, as again, E. coli has no way of knowing which direction it's going except to compare the temporal gradient. So it changes direction with the tumble. It's a random change of direction. And with some luck, it'll go away from the repellent. If not, it'll change direction again. So that's, yeah.
How does it keep the memory?
The question is, how does it keep the memory? So I'll describe that in detail this time. It's inside the proteins that make up the circuit, there's a memory.
Does it move away or towards the same speed?
Does it move away and towards at the same speed? You mean the swimming speed? So the swimming speed stays the same. The swimming speed.
The overall, the net movement towards the repellent, towards the thing that attracts it, and things and away from the things that repel. Does it end up being overall the same speed?
So the question is now, if your question is, so the speed here is, let's say, 30 microns per second, but the center of mass of this cloud moves to the pipette at maybe a few percent of that, or 10% of that, let's say a micron per second. And the speed depends on the gradient a little bit. But let's say order of magnitude, it's not that different. 1 micron per second, 1 cell length per second.
Is it linear? I mean, if the increase in concentration of the attractant is very big, will the probability difference will be bigger? Or it's just a yes, no?
So the question is, does this decrease in the probability to tumble, how does it depend on the increase in attractant? It's a kind of an S-shaped function, and we'll talk about that also. Yeah.
How do these runs, which seem much, at least in this depiction, much longer than
Tumbles.
No, the runs in terms of distance, much longer than the organism. How does that fit with what you said earlier about it being able to perceive from head to tail small changes?
So it's not able to perceive small changes from head to tail. If you put an antenna on its head to tail, it wouldn't be able to just by that know which way to go.
Population will shift just by
This is a stochastic thing. So it samples space and averages over time, you can say. And by moving like that for long times, it's able to, and using these rules, which are also stochastic, gradually bias this random walk in the right direction. Incredibly as it may seem. Yeah.
Is this in a microscope? Is there more
The question is, was this seen in the microscope? Yes, Howard Berg so he made the feedback loop that keeps this bacterium in the center of the field of view. So the microscope was challenging in the 70s because electronics didn't have very fast integration times then, but just at the barrier, like at the cutting edge of technology, was able to do it. Now it's quite easy to do. And put attractants on one side of the chamber, and you could see this increase, measure it experimentally. Yeah. And now there's really magnificent experiments of all kinds, looking at bacterial chemotaxis. It's a model system that I think every biologist has to know, and every systems biologist. It's a canonical system. A lot of it was discovered in it, and it's one of the best understood systems to go from molecules to behavior. Yeah. Okay, let's continue. I'm going to describe now an experiment that that we'll use to understand the responses. That's good for making models in theory. So a simple experiment to do is to look at response to a step addition of attractant at reactant. So here's the experiment. You take the cells. There's no gradients, no spatial gradients in this experiment. You just take the cells. They're swimming in different directions, doing the random walk. And now you add uniformly in space attractant. Okay. So what I'm going to plot here, this is time, and this is the attractant concentration. And here I'm going to plot the tumbling frequency, F. This is, I'm taking the entire population and I'm seeing what's the frequency of making these random tumbles. And without any attractant around, it's about 1 hertz, once per second. So no attractant around, and the population is. Now I'm going to add attractant. No matter which way the cells are swimming, they see an increase in attractant. So things have gotten better. So what do they do? Less tumbles. And within 1/10 of a second, they stop tumbling. Hey, let's take a nice deep sigh of relief. Good to see you. Welcome. So this is the response. Now, if I added a lot of attractant, let's say 1 millimolar of attractant, this can go on for 5 minutes. If I added just a little bit of attractant, let's say 10 micromolars, this can go on for seconds. But anyway, no matter how much I added, after a while, so they're doing random walk, and I added attractant, they stop tumbling, so they're just swimming like this, right? Maybe Brownian motion is moving them a little bit. After a while, they realize that they've been fooled. They say, wait a minute, there's no gradients. And they start tumbling again. So attractant concentration is constant. Start tumbling again. And they reach, after a while, the same behavior as if there was no attractant. So we have a response. This process is called, in biology, adaptation, which means that despite the fact that there's input signal, you normalize it out, and they return exactly to the same tumbling signal, exactly to their adaptation is precise adaptation, or I call it here exact. Exact. You can say either. Exact adaptation. So it's exactly, goes back exactly to the same tumbling frequency. That'll be important for us. Now what happens if I add, I put in another step? Response, adaptation. And I can keep doing that until I saturate the receptors till they can't, at a certain concentration they can't see any changes anymore. But as I said, there is a 10 to the 5th range where they can. Okay, so there's the properties of this experiment show us that there's a fast response, there's exact adaptation, and there's an adaptation time that can go from seconds to minutes depending on the size of the step and where you start and where you end up. This exact adaptation happens not only in bacterial chemotaxis, it happens in virtually every sensory system. For example, our visual system. Imagine a time where you go from a bright sunlight into a very dark room. And at first you can't see anything. And slowly your eyes adapt and you can see contrast again. Or vice versa, you go from a very dark room to bright sunlight. The auditory system, if your neighbor starts drilling, let's say, constantly into the wall, you notice it. But if it goes on for enough time, you stop noticing it. Smell. If there's a delicious cheesecake baking in your oven, you walk into the house, you smell it. After a while, you stop smelling it, even though the smell is still there. All this is examples of sensory adaptation. And the reason there's sensory adaptation is that it allows you to respond to further changes and also increases the dynamic range so that you don't saturate immediately and very quickly. Yes?
If the time of changes is dependent on the memory of the organism, how far back it remembers what used to be before, why are you drawing the shape of adaptation very differently from the original change? Both of them should be about losing memory. Yeah, why the two sides look so different in your graphs? Shouldn't it be
the question, why did I draw this with a shorter adaptation time than this?
No, going upwards, why would it take longer to go back and adapt to the new situation? Because all is about comparison. It's about comparing what you have now to what you had before. So the main issue should be variance in memory kept by the organism or something like that. Why is the response time to the step originally and coming back from it in different time scales? Yes?
This and that?
Yes.
Good question. Keep that question in mind. I promise you, you'll know the answer before the end of the class. Step respond exactly. What happens when I step down? What do you think? What happens when the bacteria is swimming and suddenly I take away their food? You say, hey, wait a minute, what's going on? Huh? Yeah, it's like a repellent. Going down in attractant is like a repellent. They do this. Exact adaptation.
Does it make a difference if it happens before the bacteria adapted or after?
Does it make a difference if it happens before the bacteria adapted or if I do it here in the middle?
Yeah.
It does make a difference. I don't know the answer. That's an experiment to do. Okay, so this kind of experiments are very feasible and actually done by many labs. And I did them in my postdoc. I can tell you, you can do it with image analysis of swimming cells very nicely. You can look at individual cells. And in the models we'll make, we'll try to understand this kind of response and adaptation, how it works. Good. Now, here we're talking about population averages of all the cells. If you look at individual bacteria, all have exact adaptation. But each has its own characteristic Let's call this F0. In this adaptation time, we'll call it tau. Steady state tumbling frequency and tau. So some bacteria are nervous. You look at them and they're tumbling a lot, and others are relaxed and they tumble less. So this is you see, this is what's called individuality, and this we can recognize from other organisms. They have a character, and this character lasts for a generation time. I mean, their daughters also have some correlation to the parents. It goes away after a generation or so. So it's also something to understand. Some of them are there's a diversity in their steady state tumbling frequency and their adaptation time. Interestingly, these are correlated. So if you look at different bacteria and you plot these two properties for different bacteria, there's a correlation. This is an experimental observation by Spujic and Koshland. So these are different individuals. So we'll try to understand that too. Okay. I think what we're going to do after the break is look into the cell and see the molecular circuit that does this. And we're going to take a 10-minute break now. But before we do that, let's take a nice deep sigh of relief and come back at about are genetically identical. There's no mutations, and yet there's individual character. Just like maybe genetic twins have their own individual characteristics. This response and adaptation curves, these curves appear also in psychology. It's possible to measure, for example, subjective well-being. Do you know about that? Questionnaires, very accurate measurement of, reproducible measurement of well-being. And when something happens, like you have a higher salary, subjective well-being goes up, and then it adapts, exactly, actually. And different people have different basal levels of subjective well-being and different adaptation times. There are some things that subjective well-being doesn't adapt to, like getting married and health problems. Not that I'm comparing. But a lot of the things that consumer culture is based on have to do with this adaptation. All right, so now we're going to look inside E. coli and try to understand the circuit, molecular circuit that does this computation. Chemotaxis is carried out by a circuit of 6 proteins. First of all, timescales. Up till now in the class, we talked about transcription, which is the making of new proteins, and we saw that that takes minutes, many minutes for the proteins to reach their steady state, hours. We can't use transcription circuits, feed-forward loops, etc. here because of timescales. As you saw, responses are sub-second. We have no chance with making new proteins. We can't make new proteins on the timescale that we're interested in. So we need a different level of circuitry. And here we're not need to make or break proteins, but rather modify existing proteins with rapid chemical modifications. This is an example of rapid circuits. Sub-second, second work by modifying existing proteins. And this is an example of a signaling circuit, which means it reads something from the outside and changes the state of the cell, the behavior of the cell. And these signaling circuits appear again in our cells. For example, when we have a wound, cells need to divide. They sense, let's say, growth hormones around, and they change their behavior. They divide. This is carried out by signaling networks. But there's a lot of similarity between the biochemistry in E. coli and in our cells. So biology in many ways is self-similar. You study one system in depth, and you learn a lot of generalities that apply across the tree of life. It's worth studying a system in detail. So let's look inside E. coli. I'm going to draw E. coli like this. And our goal is to understand tumbling frequency. This graph, right? When we add the step response. Again, there was a question in the break. This tumbling frequency, for physicists who are used to mean free paths, is 1 over the mean free path. You can say. So the tumbling frequency, as I said, is carried out by the flagella, the motor, going from counterclockwise to clockwise. I'm going to put it here in one end. And the signal are these attractant molecules. And the first step in chemotaxis is a special protein called a receptor. It has one end outside of the cell and one end inside of the cell. Actually, E. coli has 2 cell walls, and this is in the space between the 2 cell walls. It's a detail. So the attractant can bind here and unbind. Timescale 1 millisecond, binding and unbinding. And this receptor is what transduces the information, let's say, from the outside to the inside. Again, very general in biology. This word receptor is worth knowing. E. coli has 5 different receptors. It has 5 senses of smell, you can say. Each of the receptors can sense several attractants and repellents. But if you remove one of the receptors genetically by deleting its gene, E. coli can't make it. It can't smell. Let's say one receptor, tar, is for aspartate and ribose. Let's say this sugar. And if you delete it, it can do chemotaxis except for those particular gradients. There's 5, and one of them is for aerotaxis. It's for gradients of oxygen, actually. So there's 5 different receptors, and they all converge on one circuit that does this computation, the memory, remembering, comparing, also, which is the chemotaxis circuit. How does this work? This receptor is built like a machine. Inside, when it binds this attractant, it undergoes a conformational change, something important to know. Those proteins, in general, a lot of them, exist in discrete states. And they transition between these states on a very, very fast time scale, microseconds. And these states can be active and inactive. So in this case, the receptor is connected to a protein key A, which has I'm going to use some biology words here. That's people in biology know these by heart. People outside of biology like this word phosphorylation. Beautiful long word, phosphorylation. First time you hear it, it sounds very complicated. After a while, you get used to it. Phosphorylation and dephosphorylation. So what this key A does is it phosphorylates another protein called key Y. And it adds to it phosphoryl group. Phosphate, you can say. Now, what is this? This is a small chemical group. Let's take a nice deep sigh of relief. Thank you. And this is like a bit of information. This protein key Y, when it's attached to this phosphoryl group, changes its conformation, and it's diffusing all the time. But when it's phosphorylated, it can bind to the motor and increase the clockwise probability. So, this is just one part of the pathway we're going to talk about now. This key A phosphorylates key Y, actually takes this phosphoryl from ATP. And this key Y diffuses here. Time scale can diffuse from end to end of a cell in about 0.1 second. Again, the cytoplasm is extremely dense. So proteins aren't like in water where they diffuse maybe 100 times faster. Still, they can. And that actually probably what limits the response time, 0.1 seconds. And there's another protein here that key Z that removes this phosphoryl group. It dephosphorylates key Y. Again, proteins that add phosphoryl groups called kinases exist in all our cells. Phosphatases exist in all cells. All this stuff is the way that bits of information are passed from protein to protein also in our cells. So how do we understand this? When you add attractant, we want to understand the response, this part here, the response. So this part here, the response, we can understand. So let me draw just a small graph here. If I have the key Y phosphate, this is YP, is the key Y phosphate concentration. And here is the tumbling frequency. This is a curve that looks like this. So it's the concentration of this protein in its phosphorylated form that reads out to the tumbling frequency. F is the tumbling frequency. How many times per second you change direction, make random changes in direction.
As the concentration of phosphorylated goes up, the frequency of tumbling goes up? It should go down.
That's, I'm telling you, as the concentration of key Y phosphate goes up, the tumbling probability goes up. So when we add attractant, the question I understand stems from the fact, what happens when we add attractant? What should happen to the key Y phosphate concentration? It will go down. This is what this circuit does. This rate here, which we call A, this rate of adding phosphoryl groups is what gets controlled. You see, it's connected here to the receptor. So this A here gets controlled by the attractant. This is the signal transduction step here.
So A goes down?
Yeah, exactly. So what's going down here is this A, which is the activity of the receptor tied to key A that phosphorylates key Y. It goes down. And the receptors are built in such a way that when they bind attractant, they change conformation and become less active. It's going to be quite challenging to remember all these steps here. So get your foreheads into a... It's like a lot of minus equal plus, but that's the way it's built. Attractant binds, this activity goes down. There's less phosphorylation, but there's still this dephosphorylation. So key Y phosphate levels go down, tumbling goes down. Now attractant is around, and that's the way it would stay, right? So attractant lowers the receptor activity. Okay. So that's the way it would stay. There would be no adaptation if this was all. It would just stay down there as long as we have attractant. So where do we get adaptation from? There's two more proteins because we have 1, 2, 3, 4. So there's 2 more proteins that take care of adaptation. By the way, why are they all called key CHE? Because when you mutate them, the bacteria can't do chemotaxis. It can swim, but it can't change its direction in a fruitful way. When you delete key A, for example, there's no phosphorylation of key Y. So there's no phosphorylated key Y. Therefore, there's no tumbling. And when you delete key A, look at the cells. They're just swimming. They're just running. On the other hand, when you delete key Z, then all the Y stays phosphorylated because it doesn't get removed very efficiently. So it's tumbling all the time. Look at those cells. They're tumbling all the time. Those are the mutants, the key mutants. And there are 2 mutants that where the cells run and tumble, but they can't adapt. And those are, that's the way this was discovered, was using mutants, etc. This is the way a lot of things in biology are discovered. And in order to understand what they do, I want to tell you that the receptor, right, so one thing we learned about it is when it binds the attractant, it goes attractant binding reduces its activity. The activity of this key A connected to it that phosphorylates key Y that leads to tumbles. But there's another feature of it. The receptor has buttons on it. You can press chemical buttons and recover the activity that was lost. And those buttons are a different kind of chemical modification. So if we learned phosphorylation, here's another word, methylation. And these buttons are CH3 groups. So here's another way to add atoms to a protein and cause it to change its, going from the active to inactive conformation, methylation. And there's 2 proteins that do that, key R, that adds, and key B, that removes. So key R adds this methyl group, and key B removes. And there's 4 or 5 buttons like this, depending on the receptor. And the more methylation there is, the more CH3, the more methylation, the higher the activity. So here's a way to recover the activity that was lost when the attractant binds, is to start methylating. Indeed, if I plot here the methylation level per receptor, after I add attractant, this goes up gradually, and activity goes back up. Why does it go up gradually? It's because these enzymes are much slower. So here is this, all these steps take sub-second. These steps, methylation, take seconds to minutes. Because especially this key R is only present at about 100 copies per cell. So there's 100 versions of this protein key R molecules per cell. And it works slowly. It adds, there's thousands of receptors. And it takes time for this to build up. It's very important, this separation of time scales, because that's the memory. We add an attractant, and it's the only thing that changes in the cell is this methylation level. So the methylation level of the receptors is actually the internal memory telling us what's the attractant level outside. And E. coli constantly, even with no attractant, methylates, demethylates, demethylates, adds thousands of methyl groups along all the time, wasting energy and material, because this methyl goes out, only in order to keep the ability to have this kind of internal representation, a memory that can subtract out the external signal. So this is kind of this system is constantly in flux. Phosphorylation, dephosphorylation, methylation, demethylation, methylation, demethylation. There's fluxes. It's a non-equilibrium system. It's always in flux. And in fact, you can't do this exact adaptation and all the properties of bacterial chemotaxis in an equilibrium system. It has to be out of equilibrium in order to have all these properties. So we have
the methylation keeps going all the time. Only after the attractant induces the QI does a shift in the balance between methylation and demethylation occur?
The question is, what is it about adding attractant that causes the shift between QR and KB in order to increase the methylation rate, right? There has to be a feedback here in the system or something. So that's very, very perceptive. Exact adaptation. So exact adaptation depends on precisely increasing the methylation to balance the reduction due to the attractant. So there has to be something here that feeds back. In fact, it's known, for example, that the activity of the receptors enhances the demethylation rate. It's very difficult to understand. When the receptor activity goes down, there's less demethylation, and therefore methylation goes up. This feedback loop, I'm going to write like this. The lower activity, the lower KB rate, and therefore increase in methylation, and therefore increase in activity. See? So if I have lower activity, less demethylation, which means more methylation, which means more activity. That's a negative feedback loop. So, so this is the circuit worked out by, let's say, over 30 years by genetics, biochemistry, which biochemistry means taking these proteins out into the test tube and seeing what they do, phosphorylate each other. And genetics means modifying the DNA to change the proteins or delete them and see what happens. And there's now measurements where you can see inside the living cell proteins interacting by attaching, let's say, yellow and blue fluorescent proteins to them and looking at fluorescence resonance energy transfer, which is a quantum effect that only happens when two proteins come closer than 10 angstroms or something like that. So you can see inside living cells these interactions, very beautiful systems. So you can look inside the cell and see its brain working, you can say. And numbers here are known, the rates, this is a slow rate, as I said. This is a very fast rate. Rates are known, the halfway points of the enzymes are known, their concentrations are known, their structures are known. It's the best understood system, really. So it's a good place to do theory. Yeah.
R is not controlled?
No, yeah. So R actually happens to be working at a constant rate that isn't changing. That'll turn out to be important. It's not controlled. And the question now we can ask about robustness is, okay, what about this curve? Or can this curve, especially the property of exact adaptation, happen despite the realities of biological material, which is that the concentrations of each of these proteins, receptors, key A, key Y, key Z, key R, key B, if you take genetically identical cells in the same environment, they've just divided even, let's say, from the same cell, right next to each other, the concentration of each of these proteins varies from cell to cell by, let's say, 30%. Especially this small, low abundance protein. So, how is exact adaptation robust to changes in the proteins of the chemotaxis circuit? Or is it robust? Maybe it's not robust. And this is an experimentally testable question because you can take cells and by experimental means change the amount of, let's say, key R that they make, and then do this step experiment and see in these cells what changes and what doesn't change. Is adaptation exact, or do the cells now stop adapting exactly? And where I'm going is that this question can differentiate very strongly between different mathematical models you can write for this same circuit, and that it takes very, very specific biochemical reactions in order to make a circuit that can have robust exact adaptation. And in fact, it's those details that happen to be found in the bacterial chemotaxis system. So it's actually, let's say, the first example of a system where a detailed mechanism for robustness was worked out. Now there's many examples in different systems. Okay? Yeah.
Does the repellent bind the same receptor?
Okay. Good question. So there are 5 different kinds of receptors. There are thousands of copies of each kind of receptor. So, and each receptor has attractants and repellents. So, the one that binds aspartate as an attractant can bind nickel as a repellent. Same receptor. And there's another kind of receptor that feeds into the same circuit, let's say, that binds serine and other things. So, same receptor can bind attractants and repellents, and then different receptors can bind different kinds of attractants and repellents. Yeah, this is the circuit. And yeah.
We talked about individuality.
Yeah.
So what is the difference in the difference of the cells? Different cells with different individualities.
The question is, how does this relate to individuality? The answer will be that the difference between cells is exactly the difference between the concentrations of the proteins, in particular this key R guy. So that's the molecular explanation for individuality. Okay, so we need to start making, in order to understand questions like this, we have to make mathematical models of the circuit. In biology, you always run the danger of trying to make a model that has all the equations here, many equations with many parameters. Usually, if you start like that, you can't understand it. It's too complicated to understand. Even if you do know all the numbers. So it's very important, like in physics, to make models that are as simple as possible, but still capture the phenomenon you're interested in. That's the approach we'll take here. So you have to know what to neglect, what to keep. And then, and these lead to experimentally testable predictions. You can do the experiments, etc. One has fine-tuned adaptation. The other has robust adaptation. And the first model was actually the first mathematical model of this system made by, among the authors was Lee Siegel. He used to be here at the math department. Very important paper is a basis for further studies. And the way what they reasoned was the following. He said, I'm going to draw the receptor here. And, it can become methylated. And can become demethylated. I'm drawing a big arrow here. This is the action of R, this is the action of Z. And without attractant, it has some activity per receptor. So there's a total tumbling frequency, which is the total activity is this A0 times the methylated receptor. What I'm doing here, by the way, I'm just going to some simplifications immediately. Like I told you, every receptor has 4 or 5 buttons for methylation. First thing I'm going to do, lump them all together, just for the purpose of a simple model. The receptor can either be methylated or not. The one that's methylated is active. The one that's not methylated is not active. That's the way I model the fact that methylation increases activity. And what I'm interested in here is the total activity. That's our output. This relates to the tumbling frequency. And this XM is the concentration of methylated receptor. And this is the unmethylated receptor. And now when I add our attractant, so it's binding all the receptors. I'm going to add a lot of it just for the purpose of our simple discussion here. What does attractant binding do to the activity per receptor? Lowers it. So let's say if this is 1, just for whatever purpose, this will be 0.1. So I'm adding attractant and the activity per receptor goes down. Adaptation happens because there's more and more of this methylated receptor, as we saw. So the activity is A1 times XM. So how does this model work? What it works is, so the way it works is like this. This is the activity, which is related, let's say, equal to the tumbling frequency.
Wait,
Yeah.
what is X and what is
Thank you. This is the X is the concentration of methylated receptor. So we assume here that the methylated receptor is the active one. The unmethylated receptor has 0 activity. So we, we have, without any attractant, we have certain activity per receptor and a certain amount of methylated receptors. So we have our activity here. When we add attractant, the activity per receptor goes down. So we have here, and then the amount of methylated receptor starts going up, 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 and we're supposed to get exact adaptation.
A0 and A1 represent what?
This is the activity per receptor, and this A is the total activity of all the methylated receptors, or all the receptors, right?
So you're assuming there are a lot of receptors?
Yeah, so each receptor, as I said, is 1,000 copies of each receptor. 1,000 copies. And so it's all, you have to sum up over all of them. And this is the concentration of methylated receptor goes, let's say we start from 500 per cell and we end up at 1,000 per cell or something. By the way, in order to have exact adaptation, if I had a 10-fold decrease in activity per receptor, what do I need to do to the amount of methylated receptor to balance it? 10-fold increase, right? in order to balance this reduction. So exact adaptation means that xm increases to balance decrease in, from A0 to A1, this big decrease. Now, how this happens is that you notice that I drew a big arrow here for B and a small arrow here for B. This demethylation, according to the model, is lower when there's attractant. B works slower when attractant. Fine. You can say this is a kind of very simple way to include that feedback I told you about. Now, you can make this work. You can make this have exact adaptation. I'll show you. But you have to choose parameters. Okay? The reason you have to choose parameters is because you have to match this decrease to this decrease. You have to match it exactly. If you don't match it exactly, you don't have exact adaptation. That's the problem here. So I'm going to do the math for this very, very simple model, which is a simplified model of, we should say instead is a simplified model, but the bottom line is that you need fine-tuning in order to get exact adaptation. So the rate of change, I'm going to do this a little quickly, I guess, for some people. So don't worry about it. If you want to look at it here at the book, follow it in the lecture, or look at it in the video, etc. So the rate of change of this methylation is the amount of enzyme R, and this is the rate of enzyme R. This is the rate of methylation. How many times per second each of these molecules adds methyl groups. And as I said, this model uses what's known as that R works at a constant rate. And then there's the rate of demethylation. And this works according to standard enzyme kinetics. A function that we already talked about. This Michaelis-Menten function, which means basically that this enzyme B works on methylated receptors, and the more there are methylated receptors, the faster that it works, because it has to bind its target. And this K is like the halfway point, is the point at which the reaction is half maximum. This is a standard way to write enzyme kinetics. Now, it's when we reach steady state here, right? This is a steady state. There's no more change, so the dxm dt equals 0 at steady state. And now we need to calculate xm steady state. And you move this here, you get something like RVR. K divided by BVB. And so on. This I just got from algebra here. So our activity here, A0, before we add attractant, is just A0 times this xm steady state. And our activity here at the end is this lower activity per receptor times the same thing. But now B has a lower activity. So if this is VB, this is VB prime, which is a smaller activity, lower activity. This represents the feedback. When we have a attractant, key B works slower.
What changes
In the original model, it's that the attractant binding actually changes the shape of the receptor, making it less easy for this VB to work on it.
you can say in this model the A is whether it's A0 or A1, A0, A1 or A2.
So two things happen when the attractant binds. The activity goes down, and the ability of VB to attack this receptor goes down. Now, exact adaptation means that A0 equals A2, right? Which is fine. You can now tune the parameters. You can find VB and VB prime, everything that will make this work. Just to make it simple, I'll give a numerical example here. An example. So let's imagine we said A0 is 1 and A1 goes down to 0.1. Okay. And let's just say that everything, like K equals 1, RVR equals 1, BVB equals 2. I'm just putting in numbers. We don't need to worry about the units right now. So what do we have here? It's 1 times 1 divided 2 minus 1. This equals 1. So our initial steady state is 1. This A0 times 1 is 1. So that's 1 tumble per second, etc. Now, in order to keep this 1, I keep everything the same. I must set VB prime to, I calculated before, to 1.1. So it goes down from 2. It has to go down to 1.1. And let's just calculate it, because I have 1 times 1, 1.1 minus 1. So it's 1 divided by 0.1, which is 10. So XM goes up tenfold. And 10 times A1 is 10 times 0.1 equals 1. So I have to tune it. I have to, in these numbers, I have to change VB from 2 to 1.1, which is fine. I have exact adaptation. Great. But key R levels by only 20%. Let's imagine I have a 20% change in key R levels. Let's say I reduce it by 20%. So now R equals 0.8. And I do the same calculations. I get that A0 goes down to 6.3 or something like that. We can say 1 times 1, or let's say 0.8 times 1, 2 minus 0.8. So this is about 0.8 divided by 1.2. Oh, sorry, 0.63. But A2, look what happens. If I change this to 0.8, this goes down from 10 to 3 and 1/3 or something. And this goes down like this. So if I'm going to draw it here, what happens when I just change, I keep all the parameters the same. This cell happens to have 20% less key R. So instead of 1, I start at 0.6 something. I respond. I adapt. I don't have exact adaptation anymore. Exact adaptation is lost. So this calculation is just to make the point that this model, you can reproduce the experimental results. It's fine, but it doesn't have robustness. The property of exact adaptation is not robust to changes in parameters like protein levels.
Shouldn't that have been obvious to us, because there is nothing that's closing this loop on A2 without doing the exact
The question is, shouldn't this have been obvious to us? You can, you can. I mean, there's something that's not exactly closing the loop, but there is a kind of correspondence with the observation that Key B works slower when you add attractant. But this way of doing it, like without closing the loop, you can say, can give you exact implementation, but not in a robust way. There, you can also close the loop in ways that are not robust. But there's one way to close the loop, as far as we know, that does give you robustness, that I'd like to describe. So it's not only because here it's a feed-forward system, but also because that's not enough. A feedback system is not enough to give you robustness. You need a specific kind of feedback.
In the system, the memory is done by VB.
The memory, you can say, is the memory is carried by the methylation level, so it's Key R and Key B. Yeah. The methylation level is what changes when the attractant changes for a long time.
And if you need a memory for a large dynamic range, 10 over 5, 50 dB, then VB can't change discretely. You need it more continuously.
Exactly. So in fact, the methylation level varies logarithmically with the external concentration. It turns out in the actual model. So you have this dynamic range is compressed there by the biochemistry that calculates the logarithm by special. Okay, so how can we get a robust mechanism? And this was discovered by Nama Barkai, who's here at Weizmann. I think you all know her. And Stan Leibler, theoretically. And it's really much simpler than this non-robust model also, is mathematically. And the way it works is that you just look at the receptor, but you look at it with a stronger microscope, you can say, by noticing that this methylated receptor can exist in two conformations that change very quickly, like I told you. This is the active, and this is inactive. So before, what we did was we averaged them. They're microsecond fluctuations, and we said that's the activity per receptor. But in this model, you look at them, these different conformations. It's very important. And you notice that, sorry, I'm going to draw it like this, inactive. And the unmethylated receptor, it's not methylated, is of course inactive. Again, we assume there's two states of methylation. Methylation makes the receptor active. So Key R works and adds a methyl group. These conformations here are affected by the attractant. So the attractant binding really pushes you to be inactive. Okay. Here's the important assumption. Key B works only on the active receptor. This is, again, very plausible. These two conformations are physically different things. And all you need to do is this methyl group is only exposed for Key B to take it away in the active conformation. This is the key assumption.
Can you explain the three states? What's the difference between the top and the bottom two?
These two?
Yeah.
This protein Key M, this receptor, is going in microsecond time scale between active and inactive conformations. The attractant biases these probabilities by binding. Before we called it A0 and A1. That's the average probability to be in the active conformation. Now we're not going to use A0 and A1. We're going to look at these two conformations as two discrete objects. Again, this is a modeling reframe in the story that we tell, but it allows us to notice that Key B works only on active receptors. When you make this assumption, you get everything. And let's just see how this works. So now the activity is the concentration of active methylated receptors. And we put a star here. Usually we call it XM star. Is this the concentration of active methylated receptors? And in order to just see the dynamics here, we just need to write an equation for the total amount of methylated receptors. And notice that, again, it's just like the last time. It has RVR, and it has BVB. But this one only works on the active receptors. And if I want to, I can write it as a full Michaelis-Menten equation. Just like last time. The only difference is, instead of the total amount of methylated receptor, here it's only the active state. So, yeah, just for simplicity, I'm going to just write it like this. So XM star at steady state is just equal to RVR over BVB. And notice that this does not depend on attractant at all. So what's going on here? What's going on here is the following. I have a constant addition of methylation. When I add attractant, XM star or activity goes down when I add attractant. Therefore, more and more methylated receptors accumulate. When does this process stop? When the active receptors, this arrow equals to that arrow. So this process has an automatic stopping point, that this active receptor amount times this rate equals this rate. I'll write it in a different way. It's A steady state minus A. So this is the level of methylation. The level of methylation, the rate of change of the level of methylation. The level of methylation, the amount of methylated receptors here equals the integral over time of the error in activity. Just integrated this equation. Integral of the error, the activity, minus its steady state activity. So the methylation, total amount of methylated receptors, climbs up, up, up. It can only stop when this error is 0. The error is 0 means exact adaptation. And this principle is called integral feedback. Is a key principle in control theory and engineering. You take a device, you want to lead it to a certain fixed steady state, like a temperature in this room. I go to the thermometer and I say, okay, I want 20 degrees centigrade. That's the fixed level I want. There's an integral feedback device that compares the temperature now to the desired temperature. And that controls the power to the heater or to the air conditioning system. And the only way you can get a steady state is when this error equals 0. Or this level of methylation keeps changing until the error equals 0. And this integral feedback principle, you can prove in a wide range of systems, is the only way actually to get exact adaptation to steps and things like that, that's robust to the parameters of the system. For instance, if I open the door to the room, change the thermal conductivity, whatever, as long as I have this kind of control, I'm guaranteed to go back to the fixed point. So what's going on here? Look at bacterial chemotaxis, this random walk strategy with exact adaptation. And when we look at its molecules, we can see that we can make two kinds of models, one that's fine-tuned and one that's robust. How to tell the difference between these two models? To take a protein like KR, change its concentration, and see what happens. Do we get exact adaptation, or do we get not exact adaptation? And that's what I did in my postdoc when I came there. After Nama Barkai came to Stan Leibnitz's lab, I came there 1 year later. And I said, that's a great experiment to try out. So I genetically engineered E. coli cells by taking, let's say, each of the chemotaxis proteins, like KR, deleting it from the chromosome. So E. coli can't make KR anymore. It swims only straight. Adding it back on a piece of DNA, gluing it to the promoter of a gene that responds to sugar, and adding sugar, or IPTG, the inducer. And then I could change the levels of KR inside the cell, manipulate the levels of KR. And then do image analysis on swimming cells and give him attractant and see what happens. And what happens is, in E. coli, is that when you change protein levels like KR, so I'm going to see what happens when you change KR. You change the steady state activity. You change the adaptation time. But exact adaptation is not lost. So the steady state activity depends on KR. So it does change. But the property that you return to the same steady state level when you add attractant doesn't depend on the protein levels. KR, KB, KY, KZ. And that also explains this individuality I told you about. As I said, KR is the lowest concentration protein and the one that changes most from cell to cell. So different cells have different KR levels. And indeed, different cells, so different steady state tumbling frequencies, different adaptation times, but they all show exact adaptation. That's hardwired into the circuit. This principle of integral feedback is something that you can take with you because this appears many times when there's robust behavior in biological systems. At the heart of it, a lot of times, is integral feedback, which in biology, for biology, it's easy to make an integral over time. You just make some chemical reaction that accumulates. This accumulation is an integration. And if you make this accumulation equal to the difference between the steady state and what you're trying to control, you get exact adaptation built in. So biochemistry can do this quite naturally. It applies to since the Barkai-Leibler model was published in 1997, there's a wide range of systems, organs and embryonic development and all kinds of cellular circuits that have been found to use integral feedback. So again, as theorists looking in biology, if we have a system, we don't know a lot about it, but we can guess what the important function is, we have a tool to guide us to feasible models. Or experimentally, if we have a system and we can vary the parameters, we see what doesn't vary a lot, what's insensitive to parameters. We have a guess at what might be the important function or the function that's linked to the fitness, at least to the selection of this system. Of course, some systems rely on variability between cells, but some of them rely on precision. In that case, robustness is a very powerful principle. Okay. Let's take a nice deep sigh of relief.